Welcome to my channel. In today's video, we are going to do NCERT reading of chapter Cell Structure and Functions. So let's start our chapter. First, we will study about G.N. Ramachandran. G.N. Ramachandran was born on 1922 and died on 2001. G.N. Ramachandran, an outstanding figure in the field of protein structure, was the founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen published in Nature in 1954 and his analysis of the allowed conformations of proteins through the uses of the Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922 in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as the top ranking student in the BSc Honours Physics course of the University of Madras. He received a PhD from the Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on models of the alpha helix and beta pleated sheet structures that directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Cell, the unit of life. All organisms are composed of cells. Some are composed of single cells and are called unicellular organisms, while others, like us, composed of many cells are called multicellular organisms. Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schielen, German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form the tissues of the plant. At about the same time, Theodor Schwann, 1839, a German zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Shawn proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Sheldon and Shawn together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omni cellula e cellula. He modified the hypothesis of Sheldon and Shawn to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. You have earlier observed cells in an onion peel or human cheek cells under the microscope. Let's recollect their structure. The onion cell, which is typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called a nucleus. The nucleus contains the chromosomes, which turn contain the genetic material. DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nuclei are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplasts in plants and mitochondria and on rough ER. Animal cells contain another non-membrane bound organelle called centrosome which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human RBCs are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in the shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral-shaped. The organization of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semifluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. They are essentially enfoldings of the cell membrane. Typical bacteria is 1 to 2 micrometer. PPLO are about 0.1 micrometers. Viruses are 0.02 to 0.2 micrometers. And a typical eukaryotic cell is 10 to 20 micrometer in size. 8.4.1 Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram-Viz. That is, take up the Gram stain or Gram positive and the others that do not are called gram-negative bacteria. Glycocalicus differs in composition and thickness among different bacteria. It could be a loose sheet called a slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membrane structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the largest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophycean granules, and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Thank you.